Real-time fluorescent quantitative PCR, one of the most widely used nucleic acid techniques with Tachman probe hydrolysis and fluorescent signal accumulation. It can provide sensitive and real-time monitoring PCR process. However, limited by the number of fluorescent channels of PCR instrument, it is difficult to detect more than 4 to 6 targets in a single tube, and the multiple target detection often requires multiple PCR tubes, resulting in high consumption of original samples, low efficiency of batch detection, cumbersome operation, and high cost. The new generation of multiplex probe amplification (MPA) enables a quantum leap in single-tube multi-target detection. In each fluorescent channel, 6 to 7 pairs of oligonucleotide chain probes are designed for 6 to 7 targets. Each probe pair contains an oligonucleotide chain THO complementary to the target sequence and an oligonucleotide chain PCO partially complementary to THO. The combination of the two has a specific melting temperature Tm. THO is similar to Tachman probes, labeling fluorescent and quenched moieties, while PCO is unlabeled and binds to PCR product of the target sequence during PCR being enzymatically cleaved to produce fluorescent. When there is no target to be measured in the system, there is no participation of THO in PCR amplification, no cleavage by the enzyme, and no amplification curve is generated. In the melting curve analysis, the unconsumed THO and PCO formed a hybrid double chain, which emitted fluorescent because the fluorescent and the quenched groups were spatially distant. When the temperature rises above the Tm value of the double-stranded probe, unstranding occurs. The fluorescent group will come close to quenched group. The fluorescent is quenched, and then the fluorescent intensity decreases sharply to form a melting peak with a specific Tm value. When there is a target to be measured in the system, the THO probe binds to the target first during the PCR process, fluoresces by the enzyme cleavage to produce an amplification curve. In the melting curve analysis, the specific melting peak disappears or weaken because THO is consumed and there is no obvious fluorescent intensity change. 6 to 7 pairs of probes with different Tm values of melting peak can be set up in each channel, and eventually the presence or absence of the target to be measured can be determined by comparison with the curve of the negative control. With full use of 4 to 5 fluorescent channels of PCR instrument, 24 to 35 targets can be simultaneously detected in a single tube, single reaction. MPA technology has been patented in many countries, including the United States, the European Union, and China, and several ISCI papers have been published. MPA technology innovation, unique probe structure to achieve single-tube ultra-multiple detection, ensure high sensitivity and specificity, closed tube reaction of whole process, anti-pollution, convenient and rapid with a higher detection throughput. And it only requires a common fluorescent PCR instrument in the market, which is easy to carry out universally. Automatic interpretation of the results is more intelligent with the data analysis software. Based on MPA technology, Biotron has successfully developed respiratory pathogen multiplex test, drug resistance gene multiplex test, HPV, DNA, and RNA full typing test, fungal typing test, mycobacterium eucalysis typing test, thalassemia gene test, etc. And the application fields are expanding continuously. The diagnosis and the treatment of respiratory tract infection is highly dependent on the premise and rapid targeting of the causative agent. The MPA-based respiratory pathogen 1+1 test solution by Biotron covers 12 viruses, 15 bacteria, and a typical pathogen of the respiratory tract, including COVID-19, and is suitable for broad-spectrum testing of respiratory tract infections in multiple departments, covering many of the most common clinical pathogens. With fast detection, reduced reporting time, excellent performance, and reliable results, MPA is faster and more efficient than traditional methods and does not require expensive specialized equipment, more accessible than MNGS. As the only cancer that can be prevented and treated, MPV screening of human papilloma virus contributes to the prevention and control of cervical cancer. The MPV-based MPV panel test provides a comprehensive solution for cervical cancer. 15 MPA DNA panel tests can detect all high-risk MPV subtypes for more accurate cervical cancer screening. 31 MPV DNA panel tests can be used for multi-disease and multi-departmental application for broader spectrum multi-disease screening. E6 and E7 mRNA are early warning signs for the initiation of cervical lesions and have a higher correlation with cancer than DNA. The full typing test for HPV type 14, E6, E7 mRNA creates a more specific identification of persistent infection. Biotron has achieved a double indicator re-update of HPV DNA and mRNA. MPA leads the next generation of multiplex PCR technology re-update. Biotron's molecule diagnostic series of testing reagents are precise and unlimited.